So guys, let's begin with the second week of embryogenesis. Well, the second week is all about the week of bilaminar germ disc. Second week is the week of bilaminar germ disc. Well, we call it week of bilaminar germ disc because in the second week only two lamina will form. You can say the primitive ectoderm, the so-called epiblast, and the primitive endoderm, that is hypoblast. These are the two layers which will be formed in the second week, and that is why we call it week of bilaminar germ disc. So starting from where we left in the first week, so we had a blastocyst with us, right? So guys, let us say this is the blastocyst. We already know the outer layer is called as the outer cell mass. But once the implantation takes place, instead of calling it outer cell mass, now we'll call it trophoblast. We will refer it as a trophoblast. Yes, it is a cytotrophoblast and syncytial trophoblast. But let me just stick with the trophoblast, the simple term trophoblast. And let's say these are the inner cell mass. So remember that inner cell mass which are pushed to one corner by the formation of cavity inside, that is the inner cell mass. The pole on which the inner cell mass is attached, that is called as the animal pole, or that is also called as the embryonic pole. And the opposite pole is called as a vegetative pole or abembryonic pole. So that's an embryonic pole and that is an abembryonic pole. The cavity obviously inside is what? That is blastocele. The cavity inside is blastocele. So see, what is the first thing will happen here? The cells of the inner cell mass which are facing toward the cavity, the one which are facing toward the blastocele, they are the one which will give rise to the first primitive germ layer. Look at this. It's from the inner cell mass only, but these cells are facing toward the blastocele. These cells get converted into the first primitive germ layer, which is called as the hypoblast. This is the hypoblast. Well, as the name says, hypo, it's below. Hypoblast, also called as primitive endoderm. This is primitive endoderm. Hypoblast or primitive endoderm, because it is formed on the lower part, so we call it hypoblast or primitive endoderm. That is the first germ layer to appear. Make sure when you call it endoderm, you got to use the word primitive endoderm with it. What is the next thing happen here? Now once this hypoblast is formed, look at this guys, let's say this is the hypoblast which is formed already. Now what this inner cell mass will do, the rest of the inner cell mass, they organize themselves in a tall columnar fashion. They organize themselves in a tall columnar fashion and they'll give rise to this layer which is called as the epiblast. Epiblast, which is also called as the primitive ectoderm. That is epiblast or primitive ectoderm. So hypoblast was primitive endoderm and this is primitive ectoderm or the epiblast. Once this hypoblast and the epiblast is formed, hypoblast is the first one. So once this hypoblast and epiblast are formed, now what will happen? These epiblast and hypoblast will give rise to the cell. They will proliferate to give rise to certain cells which will cover the cavity above and below. These epiblast cells, look, I'm using the same color code also. Epiblast cells will give rise to the cellular layer which will encircle the cavity above from inside. First of all, this cellular layer which is derived from the epiblast is called as the amniogenic cells. This is called as the amniogenic cells. So one question here, that where the amniogenic cells are derived from? They are derived from the epiblast only. They are derived from the epiblast. And once these amniogenic cells, they line the cavity from inside, the cavity above, we start calling it amniotic cavity. This cavity here is called as the amniotic cavity. On the other hand, hypoblast cell will also give rise to a cellular layer which you can see will proliferate and will cover the trophoblast from inside like this. This layer which is derived from hypoblast is called as the Husser's membrane. This is called as a Husser's membrane. That's another question here, where the Husser's membrane is derived from. It is derived from the cells of hypoblast only. Amniogenic cells from the epiblast and Husser's membrane is derived from the hypoblast. And as soon as this Hughes membrane is formed, now the cavity below, you no longer call it blastocele. Now we start calling it what? The primary yolk sac. Make sure you call it primary yolk sac. So this is what is happening till now. I mean, epiblast is formed by the inner cell mass that is giving rise to the amniotic cells forming amniotic cavity above, and hypoblast cells forming the Hughes membrane, which will form the primary yolk sac below. Now, as we said in the beginning only, this is what week? This is the second week of embryogenesis, and in the second week, we said the mesoderm will not be formed. I mean, mesoderm is formed in the third week, that is called as the week of gastrulation. In the second week, mesoderm will form, but that mesoderm in the second week which forms is not the intra-embryonic mesoderm, that is actually extra-embryonic mesoderm. You will see it accumulating outside the amniotic cavity and the yolk sac. Now look at the picture, guys. When I'm saying outside the amniotic cavity and outside the yolk sac, but inside trophoblast, of course, that line over there, the black line over there is corresponding the trophoblast. So that is the next important step to understand, that where and how the extra-embryonic mesoderm is derived from. So once again, if I stick to the same diagram, let me use the same color code. I'm using the red and blue till now. This is the red color for the amniotic cavity and epiblast. And let me go with this blue, which is for the primary yolk sac. Right? This is how it is. Amniotic cavity, and this is the primary yolk sac. This layer here is trophoblast. So that's what we said, that outside the amniotic cavity and the primary yolk sac, but inside trophoblast, the mesoderm is formed, and this mesoderm, the one I'm highlighting here, is called as extra-embryonic mesoderm. This is the extra-embryonic mesoderm. There we go. This is the extra-embryonic mesoderm. Let me label it for you. This is the So the question here is, extra-embryonic mesoderm is derived from where? Well, it again is a recent Ames question only. The extra-embryonic mesoderm, please note, it is derived from the primary yolk sac and not from the trophoblast. The trophoblast, which is given in certain book, that is an old concept. It's the primary yolk sac, the cellular lining of the yolk sac, the primary yolk sac, that will proliferate and give rise to this extra-embryonic mesoderm. If primary yolk sac is not in the option, let's say, if the primary yolk sac is not in the option, then your second best answer would be hypoblast. So primary yolk sac is the best answer, that where the extra-embryonic mesoderm is derived from. If it is not in the option, I would recommend you to go with the hypoblast in that case. Hypoblast. Hypoblast will give rise to the Hughes membrane, then whole thing is called as the primary yolk sac. So primary yolk sac is the best answer. The second best answer is a hypoblast. And once this extra-embryonic mesoderm is formed, in this extra-embryonic mesoderm, some small cavities will be formed inside, guys. And all these cavities, then later on, they will unite with each other and form a single big cavity called as extra-embryonic coelom, or also called as a chorionic cavity. And that is next important step to understand, that if you look at this, going with the same diagram, once again the amniotic cavity, the yolk sac. As I said, there is a big coelom. If I'm putting these dotted lines to represent the coelom, there is a big coelom which will be formed inside. And by the virtue of this coelom, you can see the extra-embryonic mesoderm itself is divided in two parts. So you will see the extra-embryonic mesoderm in relation to the trophoblast, that is outside here, this one. And then we have this extra-embryonic mesoderm which is in close relation to the yolk sac and the amniotic cavity. Right? So this, first of all, the coelom here which is formed, the coelom is called as the extra-embryonic coelom. This coelom is referred as extra-embryonic coelom, which is also called as the chorionic cavity. extraembryonic coelom, or also called as the chorionic cavity. And because of this extraembryonic coelom, you can see the entire extraembryonic mesoderm is divided in two parts. Look at this. This one, the one that you see with the trophoblast, guys, this extraembryonic mesoderm is called as somatopleuric. The word is somatopleuric extraembryonic mesoderm. Please remember these terms. We'll be using these terms over and over again in the entire embryology, the words somato and splankno. That is somatopleuric extraembryonic mesoderm. And the one that you see with the yolk sac, this is called as a splanknopleuric. This is a splanknopleuric extraembryonic mesoderm. So two very, very important terminology which we'll be using again and again in the embryo, that is somato and the splanknopleuric. And please understand, when you say the word somatopleuric, the word that you need to focus on is the somato, the body wall. Never think about that the one which is outside is somatopleuric, the one inside is called as splanknopleuric. No, that's not the criteria. The somatopleuric is the one which is with the body wall part. Splanknopleuric is the one which is with the futures viscera. What is futures viscera, guys? The futures viscera is all derived from where? From the yolk sac. So this is the splanknopleuric extraembryonic mesoderm. If I may highlight it for you precisely, actually this is the splanknopleuric extraembryonic mesoderm and not the one which is covering the amniotic cavity from the outside. That is not splankno. In other words, what according to you is present in this circle and what is in this circle here? Just look at these two, guys. Now what do you see? What are the two things that you have in that circle? We got what cells? Those red color is representing the amniogenic cells. Right? So we have amniogenic cells. Amniogenic cells plus what trophoblast? That is somatopleuric. So it is inside, but you can see it is not along the yolk sac. So we call it somatopleuric. I'm just writing SO. Somatopleuric extraembryonic mesoderm. Amniogenic cells along with somatopleuric extraembryonic mesoderm, they together are called as the amnion. This is what is referred as the amnion. Whereas, if you look at this trophoblast, somatopleuric extraembryonic mesoderm, but with the trophoblast, this time it is a trophoblast, more precisely it's a cytotrophoblast. Trophoblast plus somatopleuric extraembryonic mesoderm together, they are called as the chorion. So I'm sure you all heard of these two terms, chorion and amnion. That's what exactly the chorion and amnion is. So amnion is the trophoblast with the the extraembryonic mesoderms, somatopleuric extraembryonic mesoderm with the amniogenic cells, and similarly chorion is the trophoblast with the somatopleuric extraembryonic mesoderm, the chorion and the amnion. And what else you should note here, that the more the size of this extraembryonic coelom increases, the extraembryonic coelom will keep on increasing, right? And once this extraembryonic coelom will keep on increasing, it will push the inner cell mass. And it will take a big chunk of the inner cell mass, and that remaining inner cell mass that we have inside is now called as the, not the inner cell mass I'm saying, that is yolk sac. Let me repeat my sentence. What I'm saying is that extraembryonic coelom will keep on increasing in size, and that will push this primary yolk sac, not the cell mass. And this, they will take a big chunk of this primary yolk sac out, and the remaining yolk sac, in which the lining epithelium is more of a cuboidal type, that will be the secondary yolk sac. That will be called as a secondary yolk sac. So primary yolk sac was the one which gave rise to extraembryonic mesoderm. In that extraembryonic mesoderm, the coelom is formed. And more the size of the coelom will keep on increasing, it will push the primary yolk sac and will take up the big chunk or huge chunk of the primary yolk sac out, and that will undergo atresia. The remaining yolk sac is now called as, which is smaller in size, is called as a secondary yolk sac. What you also need to notice here, guys, look at this part here. This is the portion where the coelom is not present. Obviously, you cannot have coelom everywhere. I mean, embryo need to hang somewhere. So this is the undifferentiated extraembryonic mesoderm. Undifferentiated, there is no coelom inside. And this undifferentiated extraembryonic mesoderm is called as the connecting stalk. Undifferentiated extraembryonic mesoderm is called as the connecting stalk. What is a connecting stalk? It is nothing but the future's umbilical cord. This connecting stalk is nothing but the future's umbilical cord. So this is what is basically happening in the second week, and we're all talking about inside the embryo. I mean, we'll see what will happen to the trophoblast and all. That is outside the embryo proper. But these are the changes that you need to know taking place in the second week in the embryo proper itself. So there is a formation of the epiblast, the hypoblast, amniogenic cells, Hughes membrane. Then there is a formation of extraembryonic mesoderm, formation of extraembryonic coelom, and then the conversion of the primary yolk sac to the secondary yolk sac. And one thing that you should be careful about here, that we said connecting stalk is the undifferentiated extraembryonic mesoderm. But when you look at this picture, it looks like the connecting stalk is connected to the amniotic cavity, which is not the case. It is a two-dimensional view, guys. It's not that it is connected to amniotic cavity. There's no point in connecting to amniotic cavity. It is actually connected to that bilaminar germ disc. I mean, if I try to show you the same picture, but from the side view, I mean, let's say this view is from the front, but if you see it from the side, it will be something like this. This is amniotic cavity. This is the yolk sac. And let me use the green color here. This is how the connecting stalk is. That is how the connecting stalk is present. So remember, connecting stalk is connected to the bilaminar germ disc here. So because you're seeing it from the front, so that connecting stalk is actually coming down and going behind the amniotic cavity and to the bilaminar germ disc. Because when you see that two-dimensional image, it looks like it is connected to the amniotic cavity. There is no point in connecting to amniotic cavity. Connecting stalk is future umbilical cord that need to provide the nutrition to the embryo, and the embryo proper is all that is there in between. That is, the germ disc is representing the embryo proper. So the connecting stalk, it comes like this and connected to it. So this is about what is happening in the second week. But as I said, right now we are focusing more what is happening inside the embryo proper. We need to see what will happen outside as well. The trophoblast, guys. First of all, trophoblast is having a somatopluric. This trophoblast is having what do you call it? Two layers. One is called as the cytotrophoblast, which is called as a cellular trophoblast, and then we have the trophoblast, which is made up of the syncytium. We call it syncytio trophoblast. So let me do one thing. Let me show you a series of some pictures here. Let us understand what is this trophoblast, what are the two layers of the trophoblast and how exactly it is giving rise to all those structures. So guys, let's use some pictures to understand what we just discussed in this second week of embryogenesis. Now as you can see, the blastocyst, here is a blastocyst, and it shows the implantation is taking place. If you look at this green color, the dark green color here, this is the trophoblast, and this is a cellular trophoblast called as a cytotrophoblast. Whereas if you look carefully, this light green colored here, it is showing you the syncytio trophoblast here. So this is a cellular trophoblast, that is cytotrophoblast, and this is syncytio trophoblast. And you can see it is invading into the uterine endometrium. Here is the inner cell mass, and you can see the cells of the inner cell mass which are facing toward the cavity. They are giving rise to the first primordial germ layer, and this is what? That is the hypoblast layer. The remaining inner cell mass will organize into a columnar fashion, we said, and they will form what? Epiblast. So look at the next picture here. We need to keep on looking at inside and outside simultaneously. See what is happening outside. Now you can see this cytotrophoblast here. There is a proliferation of the cytotrophoblast, and you can see the expansion of the syncytio trophoblast also. So gradually there is a lacuna which will be formed inside the syncytio trophoblast. We'll see that in the next image. But see what is happening here. The epiblast is formed, hypoblast is there, and the epiblast start giving rise to these cells called as the amniogenic cells, which will cover up the cavity above and forms the amniotic cavity. So if I take you to the next image, which is showing you the same. Now you can see the amniotic cavity is formed, right? You can see all these amniogenic cells are formed, the amniotic cavity is completely formed. And you can also notice that the hypoblast cells are giving rise to this cellular layer going down, that is the Hughes membrane, we said. And Hughes membrane, once it completes this blastocele from inside, this will be called as a primary yolk sac. So that's a Hughes membrane derived from the hypoblast cells. Please also look at this cytotrophoblast here and syncytio. Now what is happening to the syncytio trophoblast? There are lacunae which are formed inside the syncytio trophoblast here. And this syncytium, when it invade these blood vessels, now the blood vessels will pour their blood into these lacunae. Right? So you can see all these lacuna which are now formed inside the syncytio trophoblast. Let me take you to the next step. What we discuss next is that once this Hughes membrane is formed, look at this, this is a Hughes membrane. Once this Hughes membrane is formed, the primary yolk sac will give rise to the mesoderm called as extra embryonic mesoderm, and this here is the extra embryonic mesoderm that you see outside. This is the extra embryonic mesoderm. You also need to look at that there are regional thickenings. Can you see all these regional thickenings taking place into the cytotrophoblast? This basically is the preparation for the formation of primary chorionic villi. I'll discuss these chorionic villi separately as well. Primary chorionic villi, guys, the villi which are made up of only cytotrophoblast, that is called as a primary chorionic villi. So that's the beginning of the formation of the primary chorionic villi. And what you see outside happening now, that all the lacuna that we had into this syncytial trophoblast, these lacunas are now being filled with the blood vessels, the blood actually. So the uterine blood vessels, they are now eroded, and you can see the blood is now kind of poured into these, the lacuna which are present in syncytial trophoblast. Moving on, we also said in this extra embryonic mesoderm, which is derived from the primary yolk sac, we said the coelom will be formed inside, and that coelom will be called as what? Extra embryonic coelom, or also called as a chorionic cavity. That's what you're looking at right now. So this is the extra embryonic coelom here, or you can call it the chorionic cavity, which divides this extra embryonic mesoderm into the somatopleuric, that's a somatopleuric extra embryonic mesoderm, and this here inside is a splanchnopluric extra embryonic mesoderm. We also said that the more the size of this extra embryonic coelom will keep on increasing, it will actually take a chunk of the primary yolk sac out. You can see the primary yolk sac is now getting a portion of it getting detached from it, and the remaining yolk sac inside will be called as what? The secondary yolk sac. So the next two pictures that you're looking at will show you the same thing only. You can see this, the portion of the yolk sac is kind of being detached from it, and it is completely separated from the major yolk sac, and the remaining yolk sac here will be called as what? The secondary yolk sac. Apart from this, what also we need to focus at, if I just take you back to this picture here, now you can clearly see these primary chorionic villi formed by the cytotrophoblast. Can you see these finger-like projections? These are the primary chorionic villi. And when these primary chorionic villi, they are invaded by this mesoderm. Now, can you see that extra embryonic mesoderm? This extra embryonic mesoderm, once it invades these primary chorionic villi or this cytotrophoblast, we start calling it the secondary chorionic villi, which you can see in this image here. Look at this picture, guys. You can see amniotic cavity, you can see the yolk sac, the secondary yolk sac, of course, and I'm sure you can appreciate these villi are now invaded by this extra embryonic mesoderm, converting these primary chorionic villi into what? Secondary chorionic villi now. And when we have formation of the blood vessels inside this extra embryonic mesoderm, when the chorionic blood vessels are formed, that becomes a tertiary chorionic villi, which will form the functional placenta also. So look at this picture which I'm going to show you here, this one. This is the one which is again about, it's an enlarged view of the primary stem villi or the primary chorionic villi. You can note down this part here. We just take this portion, guys. It's an enlarged image of this region here. So what you can see, this is a cytotrophoblast. We have a extra embryonic mesoderm inside. This is a primary chorionic villi because it is not invaded by this extra embryonic mesoderm. You can see the lacuna which is present outside, and this is a syncytial trophoblast, right? So this is the 11th to 13th day when you will see the primary stem villi, or so called the primary chorionic villi, which comes into the picture. What happens next is this extra embryonic mesoderm needs to invade inside this cytotrophoblast. So that's what you can appreciate in this picture, right? You can see this is a cytotrophoblast here, and that's the extra embryonic mesoderm now invading into this cytotrophoblast and converting it into what? Secondary chorionic villi, which is a scenario of day 16 to day 17. So 16th day, you can note that secondary chorionic villi that comes into the picture on the 16th day. And then finally we have the tertiary chorionic villi. You can see the tertiary chorionic villi, we have the same arrangement is there, but this time the blood vessels are also invading inside. These chorionic blood vessels are forming the tertiary chorionic villi, and that is a direct exchange between the lacunar and the embryonic blood vessels. You can see these arrows which are showing you the exchange of the nutrients between the embryonic blood vessels and the lacuna which are formed inside the splanchno, or this one, what do you call it? The syncytial trophoblast. So this is the 21st day. That's the end of the third week. So if you're noting it down, end of the third week, that is a day when the tertiary chorionic villi will come into the picture. The end of the third week, that is 21st day. So primary chorionic villi, 11th to 13th day. Secondary chorionic villi, they come into the picture on the 15th, 16th or 17th day. That's the time when you will see the majority of the secondary chorionic villi. And at the end of the third week, you will see these tertiary chorionic villi, which are having the blood vessels also inside, are being formed. But please remember one thing, we have formation of the primary and the secondary chorionic villi all around the embryo proper, but not the tertiary chorionic villi. Tertiary chorionic villi will be formed at the site where the umbilical cord will be attached. The connecting stalk is there. So by the time you will have this connecting stalk is being formed, so tertiary chorionic villi is formed only in that part. The remaining chorionic villi which are present around the embryo, they will shed off, and we call it chorionic leve. The one which are present at the connecting stalk level, we call it chorionic frondosum, and the remaining are called as a chorionic leve. So what we're saying is that, talking about the villi, we said if it is made up of only cytotrophoblast, this is a cytotrophoblast here. And this is the primary chorionic villi. So here's the primary chorionic villi, which is just made up of the cytotrophoblast. And as we already discussed, we can appreciate them between 11th to 13th day. Between 11th to 13th day, most of these primary chorionic villi are formed. The second thing was that when this cytotrophoblast, it is invaded by the extra embryonic mesoderm. Let's presume this is the extra embryonic mesoderm here. Once this extra embryonic mesoderm invades this cytotrophoblast, so now we have this cytotrophoblast, plus we also have this extra embryonic mesoderm invading inside, and now we call it the secondary chorionic villi. These are the secondary chorionic villi, and as we already said, you can appreciate them mainly by day 16th day, 15, 16, 17. That's the time for the formation of the secondary chorionic villi. And the third point, that when this extra embryonic mesoderm, there is a development of blood vessel inside it. So here is the cytotrophoblast. We got this extra embryonic mesoderm. And now what else you will see? You will see the blood vessels which are present inside. So we have additional thing is that the blood vessels. So cytotrophoblast along with the extra embryonic mesoderm, and now we have the blood vessels inside. That is the tertiary chorionic villi. By the end of third week, or you can say 21st day, majority of the tertiary chorionic villi, they come into the picture on the 21st day. That is the end of the third week. And to remind you that what we said, if you look at this, this was the amniotic cavity. Let's say this was a yolk sac here, the secondary yolk sac. And here is the connecting stalk, guys. Look at this. If this is a connecting stalk. Now, initially, chorionic villi are formed all around, but in this trophoblast everywhere. But the tertiary chorionic villi, that's what we said. The tertiary chorionic villi, these one can be seen only present in the connecting stalk region here. So the tertiary chorionic villi will be formed in the connecting stalk region, because the umbilical cord will be the one which is attached to the placenta. So that's why this is the functional placenta which is being formed by the tertiary chorionic villi. And these villi, these tertiary chorionic villi, which are formed at the level of this connecting stalk, we call them chorionic frondosum. We call them chorionic frondosum. Rest of the chorionic villi which are present all around, they will leave. We call it chorionic leve. We call them chorionic leve. This is called as chorionic frondosum, which is present at the level of the connecting stalk. Although we haven't talked about the third week, we're just telling about that what will happen to the chorionic villi. We talked about the second week. So let us now talk about the third week of embryogenesis and see what will be the steps of the development. And third week is very, very important, guys. This is called as a week of gastrulation. The germ layers, the endoderm, ectoderm, mesoderm. And when I say endoderm, ectoderm, mesoderm, I'm talking about the definitive ectoderm, endoderm, and mesoderm. They will be formed in this week here. So let's understand this third week of embryogenesis, and that is called as the week of gastrulation. This is also called as the week of gastrulation. The meaning of the word gastrulation means formation of the 3 germ layer. That is called as a gastrulation. Okay, what we need to pay attention to is, now we already saw that this was the amniotic cavity and the yolk sac thing, so we don't have to focus to the entire thing as of now. Let us only focus to these 2 layers, the epiblast and the hypoblast, guys. I'm just gonna take these 2 layers out of the picture here. I hope you already remember from the diagram that epiblast layer is having these tall columnar cells, the columnar cells that is in the epiblast, right? And hypoblast cells are actually cuboidal. It's a secondary yolk sac lining which is cuboidal in nature. So hypoblast cells are cuboidal, but what happened at one end of the embryonic plate, some hypoblast cells will get elongated. At one end of the embryonic plate, you can see the elongation of some hypoblast cells. And these elongating hypoblast cells will give rise to the formation of the plate called as a procaudal plate. This is called as a procaudal plate. So once these hypoblast cells elongate at one end, I mean, we don't know which end. I mean, this, let's say there's a hypoblast here, epiblast above. So hypoblast at any end of the embryonic plate, some cells get elongated. And as soon as those cells get elongated, it is decided that this is gonna be the head end of the embryo, and the opposite will be the tail end of the embryo. This is the head end, and that is the tail end of the embryo. So in a way, procaudal plate basically is the first structure. It is a first structure to determine the craniocaudal axis of the embryo. It is the first structure to determine the craniocaudal axis of the embryo. And please be careful about this. It is the first structure to determine the craniocaudal axis. It is not the structure which also decides the right and left of the embryo also. Craniocaudal axis of the embryo as well as right and left of embryo, both are kind of decided, or you can say the lateralization of embryo can be done once something in the midline is formed. And that is actually notochord. So these are 2 different questions, guys. First structure, the first indication or the first structure to decide craniocaudal axis, yes, it is procaudal plate. But the structure which decides the craniocaudal axis as well as right and left of embryo is not procaudal plate, that will be the notochord. We'll discuss that part. That will be the notochord. Please remember that. So right and left of the embryo will be decided after the formation of the notochord. So coming back to this, so we said procaudal plate is the elongation of the hypoblast cells at one end of the embryonic plate. So what will happen in the epiblast? Now, as soon as this procaudal plate is formed, in the hypoblast, in the epiblast, what will happen? Some changes will take place, and those changes will start from the tail end of the embryo and will run toward the cranial end. That line which is formed in the midline, that is called as a primitive streak. I'll show you that in the pictures also, but look at this as of now. So what we're saying is we got this hypoblast here. We got this hypoblast, the cuboidal cells. We got some elongating cells forming the procaudal plate, right? So that is a hypoblast. This here is the epiblast. And see, what will happen in the epiblast? From the tail end of the embryo, the epiblast cells will start proliferating, and this proliferating line of the epiblast cells will run towards the cranial end. Well, never reaches the cranial end, but you can see it is running towards the cranial end and having this nodular cranial end as well. This line, which you can see is starting from the tail end of the embryo and running toward the cranial end. I mean, this, let's say this arrow is representing the direction of the growth. This is called as the primitive streak. One very, very important structure that is called as a primitive streak. Primitive streak. whereas this cranial end of the primitive streak is called as the primitive knot. We call it primitive knot. This is also called as the Hansen's node. We can also call it the Hansen's node, the primitive knot or the Hansen's node. So procaudal plate is formed, that is obviously toward the head end. Now from the tail end in the midline, this line is formed that is called as a primitive streak. It's just a proliferation of the epiblast cells in the midline that is called as primitive streak. And the cranial end of the primitive streak is called as primitive knot. That's what we said. Primitive streak, it's one of the most important event happening in the third week, guys, because primitive streak cells, these proliferating primitive streak cells will go down and they will replace the entire hypoblast. Look at this blue color. There is a hypoblast there. So primitive streak cells as on midline will come down and they'll replace the entire hypoblast and will give rise to the endoderm. And that is definitive endoderm we are talking about. Once the endoderm is formed, primitive streak cells will again come from this epiblast. They will again go down and they will lay the mesoderm above the endoderm. Endoderm is now formed, so they are also forming the mesoderm above the endoderm here. And primitive streak itself is present in which layer? It is present in the epiblast. So they are also going to replace the cells of the epiblast and will give rise to the definitive ectoderm also. That means all the three germ layers are derived from where only? From the primitive streak, and the sequence will be the first is endoderm, then it is mesoderm, and then it is ectoderm. So primitive streak, this primitive streak gives rise to all three germ layers. To all three germ layers. And as we said, the sequence will be the first to form is the endoderm, then next to form is the mesoderm, and the third or the last one to form is the ectoderm. In certain books it is written as endo, ecto and meso, which is not true. It is endo, meso and then ectoderm, which are formed by the primitive streak only, but the sequence is important. They have asked this question also, that what is the first germ layer to form, and that obviously is what? Endoderm. Now there is more to understand about this, the formation of mesoderm, guys. When this mesoderm is formed by the primitive streak, well, the mesoderm is running obviously between the endoderm and ectoderm. It is spreading everywhere, but there are two sites where the mesoderm is not able to reach. One is the procaudal plate, that is the cranial end of the embryo, and one which is just behind the primitive streak also, in the midline only, that is called as a cloacal membrane. So these will be the two sites where the mesoderm coming out of this primitive streak is not able to reach there. So those two sides are devoid of the mesoderm, and that makes complete sense that why they are devoid of the mesoderm. Let me again show it on the picture here. That will make more sense, and then we'll proceed into this. So look at this picture, guys. This is a wonderful image. This is taken from the Larson's Embryology, and what you can see in this image is you can see it from the side of the amniotic cavity. This is the amniotic cavity side, and obviously below that is the yolk sac side. The below is the yolk sac. So obviously when you see it from the amniotic cavity side, you will be looking at the epiblast, and obviously below will be the hypoblast. The blue color over there is representing the epiblast, and the yellow there is representing the hypoblast. Now you can see if this is a procaudal plate, look at that. That dotted line over there represents the procaudal plate, the cranial end of the embryo. And see from the tail end of the embryo, you can see this line which is running, that is called as primitive streak. It is in the epiblast, that is primitive streak. And the cranial end of the primitive streak, this one is a primitive knot. Now what you see, this primitive streak, the cells of the primitive streak, they will proliferate and they will go down. That's what we said, and form the endoderm and then form the mesoderm also. So let me show you another picture in which you will see that endoderm is already formed and mesoderm is being formed. So this is an image which is a junction of the two. Look at this. The same picture, but this time what you're looking at, the primitive streak. Look at these arrows which are going down here. That is a mesoderm. That is a proliferating mesoderm. And that's what we said. The mesoderm is formed everywhere. It is spreading everywhere in between the ectoderm and the endoderm, except at two sites. Look at those two circles here. The cranial end, that is called as a procaudal plate, and the caudal end, that is called as cloacal membrane. So what you see, the mesoderm which is being formed is not able to reach these regions, these sites, and that, as I said, makes perfect sense that why there is no mesoderm in that site, because that in future will rupture to give rise to the opening of the mouth, and this in future has to rupture and give rise to the opening of the anal canal. So we don't need a mesoderm there. We want the membranes to be as thin as possible so that they easily can be ruptured. Mesoderm gives rise to the bones, ligaments, connective tissue, cartilages, ligament, tendons. We don't need anything there. We just want the ectoderm and endoderm there, no mesoderm, and later on these membranes are easily ruptured to form the opening of the mouth and opening of the anal canal. So coming back to the notes, I mean, if you're seeing the embryonic plate from the top, the same view guys, if you see this embryonic plate from the top, that's the view. That is an embryonic plate, you're seeing it from the top, right? That is an embryonic plate. Here is the procaudal plate, let's say. This is the procaudal plate. The procaudal plate which will give rise to the buccopharyngeal membrane. Well, and obviously this buccopharyngeal membrane has nothing to do with the buccopharyngeal fascia. This buccopharyngeal membrane, it's an embryonic membrane. We got the primitive streak which is running from this tail end toward the cranial. That's the primitive streak, right? This is the primitive streak. And this here is primitive knot. If I mark this point as well, which is behind the primitive streak, we said this is the cloacal membrane. And this buccopharyngeal membrane and cloacal membrane, they are the two sides which are not invaded by the proliferating mesoderm. So when the mesoderm is being formed, I'm not drawing that part. When the mesoderm is coming out of this primitive streak, they are not present at the buccopharyngeal membrane and cloacal membrane. This is asked in the AIIMS exam also, that buccopharyngeal membrane is made up of what? It is made up of ectoderm plus endoderm. And same is the story for cloacal membrane as well. Cloacal membrane is also made up of what? Ectoderm plus endoderm. No mesoderm in these sides, that is ectoderm plus endoderm, that forms the procaudal plate of buccopharyngeal membrane and the cloacal membrane. So guys, once the ectoderm, endoderm, mesoderm, everything is formed, I mean, we talked about the primitive streak, but we didn't pay much attention to primitive knot or Henson's node. Now the cells from this Henson's node or the primitive knot, they will go down and they will keep on replacing the axial mesoderm. They will replace the mesoderm in the midline. First they form the notochordal process, then they form the notochordal canal, and then they form the notochord. So we don't have to go into all the steps here, but please remember that the formation of notochord is actually by the cells which are coming from the primitive knot region, primitive knot cells. So these primitive knot cells, they will go down. I mean, we are looking at from the epiblast side, we are seeing it from the above. So those primitive knot cells will go and replace the mesoderm in the midline. So it is present below in the mesoderm. This is what you call as the notochord. This is notochord. So if the question is asked, notochord is derived from where? I would say the best answer for the notochord would be it is derived from the primitive knot. Better answer than primitive streak, it is primitive knot. Now, what is the fate of the primitive streak? What will be the fate of the primitive knot? That is what we need to discuss here. What you see in the primitive streak, guys, primitive streak, it already gave rise to all the three germ layer, and then eventually the primitive streak will disappear, right? That's the rule in the embryology. Once the structure gives rise to what it is supposed to give rise to, then it disappears or it just vanishes. And same is with the primitive streak. The primitive streak will disappear, and it disappears from cranial to caudal. But sometimes what happens, the caudal end of the primitive streak may persist, and that is definitely not a good news, because if this part here, I mean, you can write it separately if you don't want to spoil the diagram. Persistence of the caudal end of primitive streak. Persistence of the caudal end of the primitive streak, and in that caudal end of the primitive streak, the primordial germ cells will come. The primordial germ cells will come into that persistent caudal end of the primitive streak, and that can give rise to the sacrococcygeal teratoma. Sacrococcygeal teratoma. Just be careful about this question. If the question is asked, the sacrococcygeal teratoma is derived from what cells? The cells are actually primordial germ cells, but where? In the persistent caudal end of the primitive streak. So primordial germ cells in the persistent caudal end of the primitive streak can give rise to the sacrococcygeal teratoma. What about notochord? I mean, what is the purpose of the notochord first of all? Why the notochord is formed? When the notochord is running in the axial mesoderm, the main function of notochord is actually to stimulate the surface ectoderm and convert that surface ectoderm into the neuroectoderm. Let me explain that to you. Okay, now look at this, guys. Look at this piece of paper. Let us say this is the ectoderm here, right? If this is the ectoderm, obviously there's a mesoderm below it, and then we have endoderm below that here, if you see it in that sequence. And we already said running in the axial mesoderm, that is what? That is notochord. Let's say this my pen here, it's a notochord here. And we said the function of notochord is to stimulate this ectoderm, the surface ectoderm, the one which is just above it, and convert this ectoderm into the neuroectoderm. And once the neuroectoderm is formed, this neuroectoderm will start to fold. And that fold will be called as what? Neural fold. So we have a neural groove in between. You can see a groove there. And then we have a neural fold on both the sides. And gradually this neural groove will keep on getting deeper and deeper. This neural fold will come close to each other. And these neural fold, they will unite. And that will give rise to a tube that is called as a neural tube. So what you can see, that neural tube is formed by the ectoderm only, right? But that ectoderm is stimulated by what? By the notochord. So notochord is not forming the nervous system by itself, but notochord is the most essential structure to stimulate the formation of the nervous system. So if I just make a small note on this notochord, we said notochord will stimulate. It will stimulate the surface ectoderm. It will stimulate the surface ectoderm and convert it into neuroectoderm. So it stimulates the surface ectoderm and convert the surface ectoderm into the neuroectoderm. That's what the function of notochord is. So notochord, after stimulating the ectoderm to form the neuroectoderm, then we only have the remnants of the notochord, right? So there are some important remnants of the notochord that we should know about. The one very popular remnant which everyone knows about is the nucleus pulposus. So one remnant of the notochord is nucleus pulposus. Another remnant of the notochord is actually the apical ligament of dens. There is an apical ligament of the dens. You know, dens odontoid process. We talked about that in the HNF also. So apical ligament is attached to this odontoid process. That apical ligament of dens is also the remnant of the notochord. And then we also have something called as membrana tectoria. That is membrana tectoria or tectorial membrane. It is just the continuation of posterior longitudinal ligament. We call it tectorial membrane. So nucleus pulposus, apical ligament of dens, and membrana tectoria or tectorial membrane, which is the continuation of posterior longitudinal ligament of vertebrae. These are the remnants of the notochord. That's an important question. The remnants of notochord, these are the three. So this is what the notochord is doing. And if we just understand this section better by taking a section exactly along the notochord. I mean, look at this picture here. If I take a section from the level of the notochord, let's say somewhere from here. If I take a transfer section from somewhere from here, let's see how that section will look like. So how that section will look like, guys? I mean, if you take a section from the level of notochord, you will see the ectoderm has started to fold, forming the neural folds. Mesoderm will be there, having the notochord in the middle, and then there is an endoderm also present below that. So this is the next section that we're going to look at. So what we said, ectoderm. It's a transfer section, guys. So ectoderm starts to fold on itself, and that is the neuroectoderm. And look at these folds. Let's say this here is the endoderm. In between, let's imagine this all is the mesoderm, right? This is a mesoderm there. And the one which has replaced the axial mesoderm, here is the notochord. Let's say this is the notochord, right? I hope you are oriented to this section. This is the ectoderm part, which you can see the folds and everything in the ectoderm. Then we got this endoderm. In the middle, that is meso. that is mesoderm. This, of course, is the notochord. That is the notochord here. And see, what are these? These are called as the neural folds. First, the folds are formed, neural tube is eventual. So these are the neural folds. And in between the neural folds, you will see the groove, which is called as a neural groove. Neural folds and neural groove. And something very, very important which will form here. If you look at the margins of these neural folds, or you can say the crest of the neural folds. So we have a very special population of cell which is formed here at the crest of the neural fold, and that's why this is also called as what cells? The neural crest cells. So one very, very important population of cell called as the neural crest cells. Why it is so important? Because neural crest cells are giving rise to so many structures. I mean, they're giving rise to an array of structures, and that is one of the reasons that many authors have started calling it the fourth germ layer also. Imagine ectoderm, endoderm, mesoderm, 3 germ layer, and neural crest cell, because of the number of structures which are formed by it, it is now also regarded as the 4th germ layer. This again could be a question, that what is called as a 4th germ layer, and that is the neural crest cells. We'll talk more about the derivatives of neural crest cells lately in this part. So what is happening, guys, that what we saw, that ectoderm is being folded, and that is forming that is going to form the neural tube. Right now it's neural groove there, and we have population of cell called as a neural crest cells. Okay. Now let me again use this paper to explain you that what is going to happen next. Now look at this, guys. Imagine if this is a neural fold is being formed, and then these neural fold obviously will fuse to form the neural tube. But it's not that the entire neural tube is formed at once. Now there has to be the site where the neural tube will fuse first, and then rest of the neural tube of the fusion will take place. So that again is a question, that where exactly the fusion of neural tube begins. Imagine if this is the neural tube, and the fusion of the neural tube actually begins at the cervical region. Not cranial, not caudal, it is a cervical region where the fusion of neural tube begins. Although it is also believed that few journals say that it's a multiple site where you can see the neural tube fusion begins. But going with the most standard text which is available in the textbook right now, that says that fusion of the neural tube begins at cervical region. So it's not exactly cranial, it is cervical region where the neural tube starts to fuse. And see, and once again think about it. I mean, if the neural tube, let's say, is fusing at the cervical region, let's imagine if this is a cervical region, somewhere here it is. If you're imagining it for me, this is a cervical region here. So the fusion of the neural tube will go toward the cranial end and toward the caudal end also. So obviously, where will you reach first? We'll reach the cranial end first, and then we'll reach the caudal end. And that is why this cranial neuropore, this opening here, also called as an anterior neuropore, we can call it cranial or anterior neuropore, will close first, and the caudal or the posterior neuropore will close later. So the fusion of the neural tube begins at the we said at the cervical region, but the cranial, or which is also called as anterior. Please remember these alternative terms: cranial or anterior neuropore will close at day 25. Well, 25, 24, all these are right answers. But once again, I'm going with the standard text in that. The cranial neuropore will close on 25th day, day 25. Whereas the caudal neuropore, caudal, which is also called as a posterior, caudal neuropore will close on day 28th. Caudal neuropore will close on day 28th. Day 24 and 27 is also the right answer. Day 25, 28 is also the right answer. But this is a more accepted answer. We'll go with that. The cranial neuropore or the anterior neuropore will close on the 25th day, and the caudal neuropore will close on the 28th day. Okay, once the neural tube is formed, what another important thing to notice is that the neural crest cells, remember that those neural crest cells present at the margin, those neural crest cells will detach themselves from the neural tube as well as ectoderm. It's a completely different population of cell. It is having so many special characteristics, giving rise to so many new things. So guys, neural tube will detach from the ectoderm. Ectoderm will replenish. So it will be something like, if I just again use these papers with me. So look at this. Let's say the neural tube is now formed here, right? Neural tube is formed. Ectoderm above it will replenish. So it's like neural tube is detached itself from the ectoderm. So ectoderm will be seen above and the neural tube will be seen below it. And then on the side of this neural tube, you will see those special population of cell called as a neural crest cells. So now we have like 3 different entities are separated from each other. So the scenario would be something like this. This is the neural tube. That's a neural tube formed now. The ectoderm is replenished. There we go. This is the ectoderm, replenished ectoderm. Neural crest cells. There we go. These are the neural crest cells which you will see on the side of the neural tube. They migrate to the different region also, but you can see them on the side of the neural tube. Here is the endoderm. That's the notochord. And let's say all this shaded region here, that is the mesoderm. So this is the notochord, the neural tube. We said the neural crest cells will come onto the side. These are the neural crest cells. We have ectoderm and endoderm. You know, already know that. This is the ecto and the endo. There is the ectoderm. This is the endoderm. And what you'll see, the mesoderm itself will also divide in 3 parts. There are 3 subdivisions of the mesoderm. Now that's the important thing coming up here. What you'll see, there is a subdivision of the mesoderm as well. So mesoderm will be seen dividing into the para-axial mesoderm. There will be an intermediate mesoderm. And there will be the lateral plate mesoderm. This will be the lateral plate mesoderm. I'll show you one very nice comparative picture also for that. So this mesoderm that you see, neural tube is like an axis. So it is just next to it. We call it para-axial mesoderm, next to the axis. So this mesoderm here is a para-axial mesoderm. This one is the intermediate mesoderm in between, and the one which is present more laterally, that is a lateral plate mesoderm. This is the lateral plate mesoderm, paraxial, intermediate and lateral plate mesoderm. So the next discussion of ours is based on what changes you will see in this paraxial mesoderm and the intermediate and the lateral plate mesoderm. So let me just give you a hint here itself. Guys, intermediate mesoderm is something which will contribute to the formation of the urogenital system. Well, not entirely, but yes, it will contribute to the formation of the urogenital system. All these genital ridge, mesonephric duct, paramesonephric duct, they're all formed in the intermediate mesoderm. We'll talk about that eventually. So that's what the intermediate mesoderm is going to do. It will contribute to the urogenital system. And then comes these two, the para-axial mesoderm and the lateral plate mesoderm. And I very sincerely suggest you, every time you read the para-axial mesoderm, you should read the lateral plate mesoderm immediately along with it. Because whatever is derived from the para-axial mesoderm, and then you will see there are certain structures which are not derived from the para-axial mesoderm will be derived from where? Lateral plate mesoderm. Now one example, like if let's say one is forming a skeletal muscle, other will form the smooth muscle. If one will form the axial skeleton, other will form the appendicular skeleton. So it's very contrasting when you talk about this para-axial mesoderm and the lateral plate mesoderm together. Para-axial mesoderm, guys, will give rise to something called as the somites, which is our next topic of discussion. But I'll tell you what is that. So the para-axial mesoderm will give rise to the somites. And what you see in the lateral plate mesoderm is going to divide in 2 parts. There is a formation of coelom inside the lateral plate mesoderm, and that will divide the lateral plate mesoderm in 2 parts. One will be the somatopleuric lateral plate mesoderm. I'm just writing SO, somatopleuric lateral plate mesoderm. And the one will be the splanchnopluric lateral plate mesoderm, SP, splanchnopluric lateral plate mesoderm. We use these words somaton, splanchno with the extra embryonic mesoderm, somatopleuric, extra embryonic, splanchnopluric. Now we are talking about the intraembryonic mesoderm, this intraembryonic inside the embryo proper. That lateral plate mesoderm is going to divide into the somatopleuric layer and the splanchnopluric layer. We'll see that. So let me first show you an image here in which you can correlate that what you just drew. I wanted to have a view of this diagram one more time, and then try to compare it with one real-time section which I'm showing you from the atlas. So this is what you're looking at right now, isn't it? So what you see in this picture here, that ectoderm is replenished, right? This ectoderm is replenished. Endoderm can be seen. You can see that endoderm there, right? This obviously is what? That is a neural tube. That is a neural tube. This is what? That is notochord. Don't worry about all these images. You'll get all these images as a separate file in your app only. So just don't worry. So there is an ectoderm. There is an endoderm. This is a notochord. That is a neural tube. And then look at this mesoderm here, guys. The mesoderm is divided in three parts. This yellow color here is telling you about the paraaxial mesoderm. That is just lateral to the next to the axis. That is paraaxial mesoderm. This small one in between it, what is that? That is intermediate mesoderm. And if I enlarge this for you, this is the lateral plate mesoderm, which you can see is divided by the coelom. What was the coelom? It's an intraembryonic coelom this time, not extraembryonic. It is inside. So it's an intraembryonic coelom dividing the lateral plate mesoderm in two parts. Somatopleuric lateral plate mesoderm, which is above. Splanchnopluric lateral plate mesoderm, which is below. Obviously, the one with the endoderm, or the one which is facing toward the yolk sac, that is splanchnopluric, with the viscera. And this is somatopleuric, it is with the ectoderm, that is with the body wall, that is the somatopleuric lateral plate mesoderm. What we also said, that paraaxial mesoderm, this paraaxial mesoderm will give rise to what? Somites. Now, if I just show you one more picture here in which you can see how the somites will look like. Now, the next picture I'm showing you, you will view that picture from the top. If I remove the ectoderm, guys, just imagine it, because once you imagine it, it will be better to compare it with the real-time section. So if you see it from the top of the embryonic plate, the layer above is ectoderm. If I remove the ectoderm, then what I'll see in the mesodermal region, I will see the neural tube which is present here, because neural tube was detached. So in the midline, you will see neural tube, and on both the side of the neural tube, you will see the paraaxial mesoderm. And in that paraaxial mesoderm, you will see some ball-like condensation present throughout, and those are called as what? Somites, which will look something like this. Look at this image. In this, what you can see, that we have this ectoderm is being removed, right? Isn't it? That you can see this torn ectoderm. So you can see the neural tube in the midline. On the side of the neural tube, these are the paraaxial mesoderm. On both the sides, that is paraaxial mesoderm. And look at this ball-like condensation in the paraaxial mesoderm. This is what you call as what? These are the somites. These are the somites. And that is our next topic of discussion as well.